In this lab experiment, we set out to compare the amount of microplastics found in different bodies of stagnant and moving water. We hypothesized that the stagnant water will have more microplastics because it is moving and therefore less likely to filter out any microplastics in the water or deposit them on the shore. In bodies of water, such as rivers and streams, the movement of water through different rocks, sediments, and leaves could filter the water of any microplastics or other contaminants, and the currents could wash them up on the shores. To test our hypothesis, we collected samples from St. Mary's College Pond, a creek, the Elk River, and a pond. For this lab, we used a Buckner funnel, Buckner flask, filter paper, a vacuum seal, suction, a rubber tube, 4 150 milliliter beakers, 4 petri dishes, ethanol, and a digital microscope. First, we poured 40 milliliters of each sample into 150 milliliter beakers. Next, we put about 3 pipettes worth of Nile red dye into each sample. Ooh! 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 Let the dye sit for 1 hour and stir every 5 minutes. Here's a diagram of what the Buckner filtration system looks like that we're going to use. We're going to pour ethanol on top of the filter paper when the suction starts in order to get the seal, and then we'll pour our samples through the filter paper in order to separate the microplastics out. Here's a video of me using the Buckner filtration. So I'm pouring in the first sample right there. You can see the liquid is getting sucked through, and hopefully the filter paper is keeping any microplastics from getting into the beaker. Pouring slowly just so that it doesn't get overfilled or anything, doesn't overflow. And then I'm going back in with some ethanol to rinse out the beaker just to make sure that there's no small microplastics left in there. Here is what's left of our samples after we put the liquid through the Buckner filtration system. So we just have the filter papers on petri dishes, and then we are going to use the microscope to look at them. We are slowly rotating the petri dish and using a blue light in order to see the microplastics under the microscope. Here's an image of what the microscope was showing us. It's kind of hard to see, but there are some dots on there that showed up as microplastics.
The 4 different bodies of water resulted in a variation in the number of present microplastics. The Duck Pond, stagnant, had 11 pieces of microplastic. St. James Pond, stagnant, had 2 pieces. The river, flowing, had 3 pieces, and the creek, flowing, had 4 pieces of microplastic. The results were inconclusive. We theorized that bodies of water around populated areas tend to have more microplastics due to increased littering. Our sample size was also small, with 4 samples from different parts of Maryland. The stagnant water samples had the highest as well as the lowest number of microplastics, and the duck pond had such a higher number than the rest of the samples that it could have been an outlier. We conclude that there is not enough data to support our hypothesis. The results were inconclusive because there was not a substantial amount of data. Unfortunately, the sample size was not large enough to accurately measure whether stagnant water would have a larger amount of microplastics than flowing water because of the levels of filtration between the two types of water. To better test our hypothesis, we conclude that we would need to run another experiment with about 100 samples from a wider variety of locations across Maryland, including samples taken from the same bodies of water at different places and times.